Now my topic is concept of biotechnology. Biotechnology, we are using biotechnology since time immemorial. So making card at home, wine making, all these are part of biotechnology. So what is biotechnology? Biotechnology are the techniques of using living organisms. It can be entire living organisms like microorganisms, plants, animals, or cells, tissues, any biomolecules that is connected with living organisms. And these are used to produce products and processes which will help the human beings, which will be useful to human beings. Now, he is Carl Erecki. He is called as the father of biotechnology. He coined the term biotechnology in the year 1919. He was an Hungarian agricultural engineer who explained and defined biotechnology. Now let us see what is modern concept of biotechnology. Modern concept of biotechnology, it includes genetic engineering techniques that are used to develop products and technologies for human welfare. So as per modern biotechnology, genetic engineering tools are an integral part of biotechnology. So as we are developing DNA vaccine or any sort of gene modification or correction, in vitro fertilization, genetic engineering techniques are used. Now, what is the definition of European Federation of Biotechnology? It is integration of natural science and biological science, molecular analogs for products and services. So now, principles of biotechnology. There are two principles. First one is genetic engineering and the other one is bioprocess engineering. What is genetic engineering? Genetic engineering, it includes set of technologies that are used to alter the genetic setup, that is DNA of an organism. Why the DNA needs to be altered? DNA needs to be altered because to introduce some new traits and to produce some useful products. So new gene that will be introduced, that will introduce some new traits and new products will be produced for that. Now, for genetic engineering, we are indebted to two scientists. He is Herbert Boyer and he is Stanley Cohen. Herbert Boyer, contribution of Herbert Boyer is, he for the first time used restriction endonucleases, and he also isolated one restriction endonuclease, EcoR1, from Escherichia coli or E. coli. We will study about restriction endonuclease in the next slide. And Stanley Cohen, he isolated plasmids and used them as vectors in recombinant DNA technology. And other contribution of Cohen and Boyer is they developed rDNA technology, that is recombinant DNA technology, in the year 1973. Now, restriction endonuclease, they are nuclease. Nuclease are enzymes which degrade nucleic acid, DNA and RNA. Now, what is the function of restriction endonuclease? Restriction endonuclease recognize specific site on the DNA and cut the DNA at those specific sites. So they'll work like molecular scissors. Like this is the entire DNA. The restriction endonuclease will study the entire DNA strand, and then it will cut the DNA at certain specific restriction site, and DNA fragments of specific length will be generated. Now plasmids. These are plasmids. Plasmids are circular, double-stranded, extrachromosomal DNA present in some bacteria. How they are used? Cohen has used them as vector in rDNA technology. Now let us see what are the different steps of genetic engineering. Now genetic engineering, first step of genetic engineering is isolation of desired DNA. Now what is the desired DNA? Desired DNA is that DNA which will code or express some useful product. So first thing there is we have to identify the cell with that particular DNA. responsible for expressing some useful product, and that DNA will be isolated, and next step that DNA will be cut at specific site using restriction enzyme, and then that DNA will be inserted in a plasmid. The plasmid will also be treated with restriction endonuclease to generate gap, and in that gap the fragment of this desired DNA will be ligated with the help of the enzyme ligase. And so now in this way the recombinant DNA is produced. Now recombinant DNA is the plasmid with the new DNA is the recombinant plasmid or recombinant DNA. Next it will be introduced, it will be transferred to a host cell, mostly any bacterium. In the host cell will multiply, and along with the host cell the rDNA will also multiply. Now what will happen? What will happen if it is multiplying? As it multiplies, it will express the protein in a much higher quantity. Here I have used the example of insulin. This DNA was responsible for expressing the protein. Now it is inserted in the plasmid, and in the plasmid it will express the same protein in a much higher quantity. So once the successful amount of protein is produced, next the protein will be harvested. Again, as it is crude, it will be purified, and now this final purified product is ready. Now let us see about bioprocess engineering. Bioprocess engineering is the process by which GMOs, what are GMOs? The organism with a new genetic setup, with a new gene, is a GMO as it is genetically modified. That organism will be grown under controlled conditions to obtain large quantities of the product. Now let us see how it is done. The organism will be grown in a bioreactor. So this is a bioreactor, and this organism is cultured on a media, and that media concentration is standardized, and then it will be transferred to the bioreactor or a fermenter to support large scale production. And this step is the most crucial, as several physical factors need to take care of. Physical factors like temperature, then pH, concentration of dissolved oxygen, all these need to be optimized, that will support maximum production of the product. Once maximum production is achieved, next step is separating the product from other substances, media and other things. So this is downstream processing. After downstream processing, the purified product is recovered, and then after downstream processing, the product is purified as it is crude, and then after purification the final product is ready. So what are the important points to remember in bioprocess engineering? First is sterility. As the microorganisms is cultured on media containing carbohydrate, so there are chances of contamination. So contamination cannot be afforded. It will be an entire loss. So sterility needs to be maintained strictly. Second point is optimization. Optimization of the physical parameters like pH, temperature, dissolved oxygen, so that maximum production will be achieved. Scale up. As it is a lab to industry technique, the GMO was grown in a lab, and then it is transferred to a bioreactor to support large scale production. So scale up is very much necessary. Along with scale ups, cost effectiveness should also be taken care of. It is also important. The entire process should be economically viable, and at the same time it should be sustainable. It should be environment friendly, and the process must be continuous. Then there is the regulatory compliance. All the rules, regulations, ethics, bioethics need to be followed strictly without compromising the quality of the product. Now, so when we are talking about genetic engineering or bioprocess engineering, it shows integration of biology and technology for large scale production of the bioproduct. I hope modern concept of biotechnology, which involves genetic engineering and bioprocess engineering, is clear to you. So more details we can study in our next lecture. Thank you.